Hello, can you guys hear me? Give me a thumbs up or something if this is working. Hello, can you guys hear me okay? Just give me a like a thumb emoji or something, or a yes. Perfect. Thanks, Ultimate Revision. The boss. Cool, we've got multiple people. This is good. How is everyone this evening? What have you guys been up to today? Hi from London. Sabi Sachi. Hello, hello. Welcome back, people who've been to these classes before, and welcome for the first time, people who are joining us today who haven't come to previous classes. You've just finished class. What, you mean your like school lessons, Sharifa? I'm guessing, are you doing like distance learning at the moment? Or are you not in the UK potentially? Quick show of hands, who's joining for the first time? Who's a new student here, or have we got mostly return people? Lennon, first time. Hannah, first time. Trinidad and Tobago, that's exciting. Jamaica, nice. A diverse crowd here. Evan, you were here last time. Good. Good. Yeah. There's a competition, exactly. So let's make a start. So wait a couple of minutes to let people give people time to log in. But yeah, I think people have had a chance now. So let's begin. I'm just going to share my screen. So, oh, where's it up there? Yeah, basically I skipped ahead a slide already. The class tonight is going to be on microscopy and sort of looking at orders of magnitude, working out how to calculate between centimeter, millimeter, that sort of thing. So really useful skills, very sort of like core skills you need for GCSE and also beyond. This also crops up at A level. And even though you learn it at GCSE, I still have some A2 students who are like, they get this stuff muddled up. So it's really, really useful to crack this. The sooner you do it, the better. So just a little bit of background. My name's Alex. I'm the head of biology here at SnapProvise. I've been teaching biology for 4 or 5 years now. Yes, the mic is better today. Those of you who were here last week, you may remember there was some issues with the microphone. I'm still not sure what was wrong with it or what was wrong, but it seems to be better today. So why are we doing these classes? It's kind of to give you a taster of what SnapProvise is about. So it's a sort of revision and learning platform. We've been running for a few years with A-level, and we're now starting to branch into GCSE classes. So due to demand. So what we're doing on YouTube today is an example of what we would normally be doing on our website. So we've got a subscription-based package. We're going to be running 4 classes a week. We cover all of the curriculum and for all the specs. So if you do one of the like GCSE specifications, it's going to be like relevant for you. Just looking at your thing, Callie, I think the downloads are only for PDFs. So yeah, you can only download a PDF version. So we also have handouts, sort of like cheat sheets, very condensed notes, really useful for revision. All the past recordings are left there. So if you ever miss a session, no worries, you can watch it at any point. And there's also some one-on-one support. So you can be asking questions directly to tutors. So if you watch a video or you go to Webclass, still not quite sure on a topic, you want a little bit more support, then that's available too through our subscription. So it's a little bit different to what we do on YouTube. So this is, who was talking about it? I can't remember now. Oh, Ultimate Revision was talking about competition. We are running a competition this month. In celebration of the launch of the GCSE package, we are giving away 10 free accounts. So all you have to do to be in with a chance of winning this competition is go to this link. Oops, where's my pen? Not drawing. Oh, it's still not drawing. Strange. Oh, it's on laser pointer. It's only letting me choose laser pointer. It's very strange. Sorry, I've never had this this issue before where it won't let me draw with a pen. Okay, we'll see if that fixes itself. If not, then I would have to maybe restart the PowerPoint. So I believe you can enter as many times as you like. I don't know if they're going to screen by email. So I probably would say just enter once. Technical difficulties, yes, which is like a weird technical difficulty. I've never seen that before. So this competition ends on the 15th of February. So make sure you sign up, make sure you enter, and to be in with a chance of winning. So that's a sort of prelude. Now let's look at the class for today. Yeah, microphone getting revenge for me bad-mouthing it. So our objectives, we're going to look at milli, centi, micro, and nanometers, and look at how we convert those and do standard form, that sort of thing, just a refresher on standard form. We're going to look at magnification and resolution, the differences between those, and then look at light microscopes and electron microscopes. And then we're going to do some calculations for magnification. Hopefully we'll get all that done. It's quite a lot to do. We'll do as much as we can in the hour that we have. So these are the spec points for the different specs. They're all pretty similar. You can see these are sort of the prefixes. I believe that's the OCR one. Edexcel, you need to go down to picometers, which are one smaller, well, 1,000 times smaller than a nanometer. The other specs stop at nanometers, but Edexcel, for some reason, you have to go even smaller. So they don't tend to come up that often in biology, but they want you to know about them. And I think my pen is still not cooperating, so apologies. Let me just reopen this PowerPoint and see if that fixes it. Okay. No, still no. Okay, I'm going to close the document and start from scratch. Apologies for this. I think what maybe happened is I didn't, you know, when it says like enable editing, maybe that's the problem. That's my theory. If not that, then here we go. Enable editing. I'm really hoping this fixes it. Otherwise, I am stumped. If anyone has any ideas, please feel free to share. Yes. Okay. Thankfully, that appears to have worked. Yeah. Sorry for the sorry for the delay. Let's see what the problem was now. I must have not pressed enable editing. Anyway, we have pen. Nice. So now I'm going to throw out some questions. So basically the way this works is there's supposed to be an interactive class. So the questions are like, I'm going to throw questions out to you guys, and if you want, like, and it's good to see, if you reply, you're going to get more out of the session. So I can see your answers, check for any misconceptions. So yeah, the more you interact, the more you'll gain from the session, basically. Okay, so yeah, first one is, what are eukaryotes and prokaryotes? This is kind of like what we were looking at last week, for anyone who was there. Or if you're struggling to define them, maybe an example. Yeah, nice, Olivia. I like that one. So eukaryotes have a nucleus. Prokaryotes, no nucleus. And you've given me some examples. So E, G, haven't really stayed in the box. Oh well. Animal and plant cells. Yeah. No nucleus, prokaryotes, E, G, bacteria is a good example. But it's not just bacteria, there's other ones. Yeah, some other differences. We could maybe say smaller ribosomes. Oops, yes, and then ATS ribosomes, which is larger ribosomes. Eukaryotes have also what we call membrane-bound organelles. So that just basically means that they have organelles which are wrapped up in membrane. So things like mitochondria, chloroplasts, vacuoles, they're only in eukaryotes. Prokaryotes don't have any organelles in membranes. Membrane-bound organelles. So even the nucleus is a membrane-bound organelle. It's actually got a double membrane. So prokaryotes do have some features, but they're not in membranes. Okay, so next question. Which are larger, prokaryotes or eukaryotes? Has anyone answered this one already? Yeah, exactly, Fatima. So these are bigger. So it's not definitely, there are some eukaryotic cells that are going to be smaller than some, the smallest eukaryotic cells, probably smaller than the largest prokaryotic cells, but in general, the eukaryotic cells are quite a lot larger than prokaryotic cells because they've got so much more in them. Yeah, like the nucleus and all the other organelles. So finally, what are chromosomes? This is the last bit of our background info for this class. Mm-hmm. Yeah. Structures of DNA containing many genes. In humans, we have 23 different types, and we have 2 of each type, so we have 23 pairs, or 46 chromosomes. In humans, yeah, Amy, chromatin is the unraveled state. Chromosome is when they're condensed, and they condense to undergo cell division. But we call it chromatin when it's unraveled. Yeah, exactly, Zara. Prokaryotes don't have chromosomes, and they also have circular DNA versus linear DNA in eukaryotes. But anyway, I don't want to get too much into that because that's what we did last week, and we're going to do different stuff this week. So nice, good background knowledge. Happy we're ready to progress. So first thing we're going to look at is the orders of magnitude. So we've got lots of different measurements. They all relate to the meter. So these are all just short. The prefixes is short for meter. So a centimeter, like that. These are all millimeter, micrometer, nanometer. They're all something of a meter. Who can tell me what the symbols are for these measurements. I'll put the first one because I feel like everyone knows this one. cm, centimeter. Yeah, nice. Then what do we got? Nice. Millimeter, mm. And then, oh, trouble, another m. What do we do? I've seen someone put it, yeah, although it's not just a U, is it? It's the funky U like that. Oh, sorry, I done the U on the wrong bit. Let me draw that again. Funky U with a long line down it. So that means micrometers. And then nanometers are more easy, just nm. So that's our symbols. Now let's work out what fraction of a meter they are. So a centimeter, kind of in the name, like centurion was a soldier who was in charge of 100 warriors, I think, anyway, in Rome. That would make sense if it was 100, because it basically means 100. So a centimeter is 1/100th, of a meter. What is a millimeter then, as a fraction of a meter? We can do them for all of these. So a millimeter is therefore 1/1000th of a meter. Micrometer, anybody? Nice, Evan. Yeah, so a micrometer is actually a millionth of a meter. So 1,000,000. And a nanometers are really, really, really tiny. So there's 1 billion nanometers in a meter, which is a lot. Although I find that less crazy than the fact that in a millimeter, there's 1 million nanometers. I know it's the same thing, but I don't know, that seems more crazy to me, because you can look at a ruler, like look at how small a millimeter is, and a nanometer is 1/1,000,000th of that tiny space. So they are tiny. The things we're looking at and things we measure in nanometers are very, very small. Okay, everyone happy with that? You need to be comfortable with these and be able to convert between them. We're going to do some practice on that later, but this is just the prelude. So just to recap the basic size of cells. So there is a bit of variety, but in general, eukaryotic cells are usually in the ballpark of around 10 to 30 micrometers in length. So let's just say, label this one as 10. Plant cells are generally bigger than animal cells. Are larger. Prokaryotic cells, so this is our prokaryotic cell here. These are much smaller. So they're sort of in the ballpark of like around 1 micrometer to some of the bigger ones, maybe up to 10, approaching the size of a small animal cell. So many, many times smaller. So roughly like 1/10 the size of a prokaryotic cell. So yeah, as in you could fit around 10 bacteria. You could stack them up end to end, and that would be roughly the same length as a eukaryotic cell. Oh yeah, Maxinda, good point. For people who are doing EdXL, there's also Pico, which I actually, is it PM? Let's just quickly double check on the spec. Oh, it doesn't say. I actually have not used picometers, I think, ever. I don't usually teach EdXL. I just noticed it on the spec as I was flicking through before. So I'm not 100% on PM. You'll want to fact check that one, but it's 1,000 times smaller than a nanometer. So it would be 1,000 billion or trillion even. So it's 10 to the 12. Yeah, nice. Okay, it is PM. Okay, thanks for checking Yeah, it's not used very often in biology. I think in physics, maybe you use it more often, and maybe even chemistry. But we tend to stop at nanometers in biology because that's what we need. Anything smaller than a nanometer, we can't even really see it with a microscope, even an electron microscope. So we don't really worry about it. Okay, so what is standard form and what do we use it for? I'm sure you guys are familiar with it. Use it for maths and other sciences. Yeah, exactly. So we use it for very big or very small numbers. And we use powers of 10 to mean that we don't have to write out all the zeros. So it just makes it neater to read and easier to interpret data. Rather than having to like count the zeros, we can straight away see that's to the power of 7 or that's to the power of 9. And we can do comparisons much more easily. Use for really big numbers uses powers of 10 to make them smaller. So let's do an example of how we'd interpret this. E.g. 1.5 times 10 to the negative 5. What is that? So that's in standard form. What is that number in normal form as an actual number? What would that look like? And how do you work it out? Yeah, nice. Most of you got that one there. So basically, this to the negative 5 means we need to move the decimal point 5 spots to the left. So let's go 1, 2, 3, 4, 5. So we put a decimal point here. We don't need these ones. And then this is our answer, 0.000015. So these are the same. But this one's a little bit neater and easier to read than this one. To the negative 5 isn't so bad. That number's not too messy. You can kind of read that relatively easily. But sometimes we're doing things to like the negative 27, which would then be a ridiculously long number. So then we have to use standard form really to get any sort of meaning. So let's just work out what these prefixes, what power are they to? So for example, let's do centimeter together. We want to know what this would be in standard form. We need to move the decimal point 2 times. 1, 2. Then we'd have 1 times 10 to the 2. That would be centimeters. Sorry, to negative 2. So what about for millimeters, micrometers, and nanometers? What would we do? Nice, Evan. That was very quick typing. Yeah. So this one, 1, 2, 3. 10 to the negative 3 is millimeters. Micrometers, 1, 2, 3, 4, 5. Wait, sorry, 6. I was thinking that sounded wrong in my head. 10 to the negative 6, and the nanometers to the negative 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah, that's a good way to remember, Evan. So after millimeters, it's then everything jumping up 3 at a time. So you sort of times or divide by 1,000 to get between them. So what I would say is basically never use centimeters when you're measuring things in biology. In fact, let's just cross them out because it's just going to confuse you. It's much better to measure in millimeters and then you're just converting by 1,000, because to go from millimeters to meters, there's 1,000 in that. So everything you times by 1,000, or sorry, you divide by 1,000 to go this way, or you times the other way. But then everything goes up in thousands. So it makes it really easy. If people measure in centimeters, they forget that you have to convert to millimeters first. And I would just always highly recommend just using millimeters. I even made, when I was teaching in a school, I made my whole class like, I know, I think actually got the school to buy them for them, like rulers that only had millimeters on them so that there was no confusion. Because I don't know, in the heat of an exam, when all the pressure's on, like people make silly mistakes. So yeah, I recommend buying a ruler that only has millimeters on it to make your lives easier. It's like a 20p investment. It's probably worth it. Okay, so let's look at some conversions. We need to know what we multiply between to get between them. So yeah, method. By the right factor of 10. So let's work our way from here. So these are all the same measurement. 20,000 nanometers is the same as 20 micrometers, is the same as 0.02 millimeters, is the same as 0.002 centimeters. So how do we get between these numbers? Yeah. So going the top way, we need to be dividing. So divide by 1,000. Divide by 1,000. Divide by 10. The odd one out. Boo, centimeters. And the other way, we're just timesing. Times by 10. So this often gets students confused, like, do I divide or do I times to work it out? Just always like take a step back and think about it. You're going from a meter to millimeters. You're going from a big unit, a meter, think of like a meter stick, down to millimeters. They're very small units. If you're going from a big unit to a small unit, you're going to need to be multiplying. There's a lot of small things inside a big thing, basically. So whenever you're going from a large unit to a small unit, you're multiplying. And whenever you're going from a small unit to a larger unit, you need to be dividing. So yeah, don't panic. If you can't remember, just have a think. Think about millimeters and meters and how would you convert between those two. So let's take an example. We've got a bacterial cell that is 4 micrometers long. And we've got a plant cell that is 0.02 millimeters long. Which of these is bigger, or how many times bigger? J, yes, exactly. So how do we do this? We can't directly compare. Well, maybe you can think of it in your head, but it's easier to basically convert these to the same units. So we've got millimeters and we've got micrometers. This is quite a small number, so let's convert them both into micrometers. Well, the bacterial cell is already in micrometers, but let's convert our plant cell into micrometers. So what is this in micrometers? Evan, nice, yeah. 0.02 millimeters is how many micrometers? We need to, are we dividing? It's definitely 1,000. So milli to micrometers, we know it's 1,000. We're going from millimeters to micrometers. We're going to times by 1,000. So 0. Oh, it's actually the same numbers that are here. 0.02 times by 1,000 equals 20 micrometers. Therefore, we've got plant cells 20, bacterial cell is 5, 20, sorry, 40, 20 divided by 40, 4, what am I doing? Let me just start that again. The plant cell is 20 micrometers, the bacterial cell is 4, so we divide that, it's 5 times larger. Nice. So, you guys happy? Happy with that? Yeah, you can give me a number or an emoji. Nice. Mostly, that's good. So let's do some past paper questions. So what we normally do, yeah, I can go back quickly if you want to take a screenshot or something, or should I go over it one more time? So the question was, how many times bigger is the plant cell than the bacterial cell? The bacterial cell is 4 micrometers. The plant cell is 0.02 millimeters. To compare these, we need to convert them to the same units. So we just converted the millimeters to micrometers for the plant cell. This one's already in micrometers, so we don't need to convert them both. We just convert one to be the same as the other. So 0.02 millimeters equals 20 micrometers. So now they're the same units, we can compare them. How many times bigger is 20 than 4? Well, we just divide 20 by 4. It's not actually very clear, is it? Let me just write that a bit neater. And that gives us our answer, 5 times larger. Hopefully that makes sense. So let's do an exam question. A red blood cell is 8 micrometers in diameter. A bacterial cell is 40 times smaller. Calculate the diameter of the bacterial cell. So what are we going to do here? Nice, the boss. People agree? .2 micrometers. So how do we work that out? We just take 8 micrometers, divide by 40 equals 0.2 micrometers. And that's the answer. Nice. Oh, seen a whole flurry of answers come in. Perfect. Well done. So yeah, not too tricky, that one. Let's do a slightly more challenging one now. So this table shows the number of different components found in the blood of a healthy person and the blood of 2 other people, presumably not healthy. We've got the components in the blood, red blood cells, white blood cells, and platelets. And we've got, this is our healthy person. Then we've got person A and person B. So you have to calculate the difference in the number of white blood cells per decimeter cubed. So number of white blood cells, decimeter cubed of blood, between healthy person and person A. So let's orientate ourselves on the table. We're looking at number of white blood cells. So we're looking at this row and we're looking at these 2 people. So these are the values that we're comparing. So we want to know the difference between these two numbers. So 7 times 10 to the 9 minus, well, you can do it either way around really, 5 times 10 to the 10. So you can plug these into your calculator and do it directly. So I would recommend learning how to do that on your calculator so you can key in standard form and do things to the power. If you didn't want to do that, you can just think about, you could type it out. So this to the 9 is 7 billion basically. That, and to the 10 is 50 billion for this one. So we want to know the difference between these two numbers. So you could minus it this way and you'll get a negative number, which is fine because it doesn't matter whether it's negative or positive, that's going to be our result. But I would find it conceptually easier to think about subtracting the smaller number from the larger number. So let's do 50 billion minus 7 billion. What is our answer? Oh, I've seen some people have got it there already in standard form. Yeah, nice. So our answer is 43 billion, like that, or it could be negative, depends whether you, which way around you did it, which if we wrote that out in standard form would be 4.3 times 10 to the 10. And that's our answer. So hopefully that makes sense. And you can see why we use standard form, because it makes it much easier. Like you could just type this straight into your calculator. You don't have to worry about typing in zeros and typing the right amount. You can just plug that straight in and you get this result directly. So well worth being comfortable with and knowing how to do. Okay, next up, let's look at microscopes. Or just to check, is everybody okay with that question? Anybody want me to go over it again or makes sense? Cool, perfect. So yeah, let's look at microscopy. So probably the two most important words in microscopy are magnification and resolution. These get muddled up. Could you guys define what the difference between magnification and resolution is? Yeah. Yeah, so like how many times larger it is. Mm-hmm. Mm-hmm. Oh, and images than the object. Do. And then yeah, the resolution is kind of like the quality of the image. Yeah, if you don't have a good resolution, you can't really see anything, and that's because you can't distinguish between 2 points. That's kind of how we actually define it. So is the ability to distinguish detail. Distinguish between 2 points and see them as separate. And to be honest, it's the resolution that's actually the more important one, because you can magnify something as much as you like. And I'm sure you guys have seen this when you know, if you like, I don't know, say you accidentally copy a thumbnail of an image rather than the actual image, which is a very low quality version of it, and you try and magnify it, it's like horribly pixelated. and you can't really see what's going on. That's because it's got a very low resolution. A thumbnail is just like a very small version of the image. It's saved with a low amount of data, therefore a low resolution. If you then magnified it, you can magnify it indefinitely, but you're not going to be able to see what's going on. It's just going to be like a blurry mess. So we can magnify things as much as we like, but after a while, the resolution isn't going to be there anymore. So yeah, this is the key one. This is the important one that we focus on. So, and these images sort of show. So this has been magnified. This is showing low resolution, and this is high resolution. And you guys are familiar with this now with like technology and stuff. When we say we've got like a high definition TV, what that means is more resolution. So we can see more detail. The image looks crisper and clearer, and we can see what's going on. So see them as separate. So like on this picture with a high resolution, we can see where the dart ends and where the point of the dart is going into the board. At this low resolution, everything's pixelated. So we just see the different pixels. This image is also made of pixels, but they're so small because the resolution is higher, we can't see where they are. So we can see where one shape is and one shape isn't. So we could see features. So let's look at how we calculate between these. So has anyone seen this pyramid before? Or do you remember the calculation to do magnification? Yeah, CRISPR. Although CRISPR is also another thing to do with biology, with gene editing. Or is that what you were going for? Yeah. So magnification is image size divided by actual size, or maybe some of you guys call it real size. So let's fill in this table. So I just always remember I am. So you draw out your pyramid and remember I am to fill it in. And then we just put a multiplication sign there. So this can tell you all the different calculations you need to do. For GCSE, you mostly just do calculating the actual size, but you can be asked to do other ones as well. Oops. So yeah, we want to do magnification. So we can use our pyramid to calculate what that is. So magnification equals image divided by actual size. Sounds like some of you are calling this real size. And if that's what you've been taught, that's fine. Like I'm more used to calling it actual size, but if that helps. But it is basically the real life size. Oh, Evan, yeah, okay, it was a joke. I thought it was because it was the capitals. Yeah. Nice. I like a good biology joke. So let's do this calculation. What are we looking for in this one? We've got a cell. We've got its image size, although I'd say it's probably not 3.75 millimeters on most of your guys' screen, but let's just pretend that it was. And it's been magnified 250 times. So this is magnification. This is the image size. So we want to work out the actual size of this cell. So actual size is here, like how big it is in real life. We do image size divided by magnification. So image size is 3.75 millimeters divided by magnification 250. So what we might want to do before we divide it is convert it to micrometers. So millimeters to micrometers, we times by 1,000. So we get 3,750. Yeah. Then 15. Nice. Was it 15 or 1.5? Let me get my calculator. I say calculator. Definitely just using my phone. Wait, let me unlock it. Okay, I'm in. 3,750 divided by 250 equals 15. Nice. You could also left it in millimeters, which some of you did, I think, Amy, yeah? 1.5 times 10 to the negative 2. Perfectly valid answer. Yeah. 0.015 or 1.5 times 10 to the negative 2 millimeters. You can definitely leave it in millimeters, that's fine. Unless the mark scheme actually specifies and it asks for a particular unit, you can give it in whatever units you like. But I generally, the sort of thing I'd recommend is put it in the units that make the number, like, I don't know how to describe it, like the nicest number, like something from between like 1 and 100 is like a nice number to look at. So you can either use standard form or you can change the units. So I think 15 micrometers is a nicer number than 0.015. It's just sort of easier to interpret. So that's why I did it in micrometers. If that makes sense. It's not very, I don't know. It's not like necessarily wrong. It just looks neater. So let's look at the two different types of microscope. So we've got light microscopes and we've got electron microscopes. So these light microscopes obviously use light. And they use little mirrors to focus the light. So there's mirrors in here, or lenses, I should say, bits of glass, and there's lenses in here. And they're used to magnify the light. So the maximum resolution is around 200 nanometers from a light microscope. Res is 200 nanometers. So anything smaller than that, you're not going to be able to see what's going on. Yeah. So electron microscopes, these use beams of electrons. So light, it's kind of, if we take light for granted, or I guess if you're a physicist, maybe you don't, I don't know. But it's just like lots and lots of photons that are moving in waves. Electrons do much the same thing. They're tiny particles that move in waves. They actually move in much smaller waves. So a photon, for photons moving like that, an electron is moving like that. So they've got a smaller wavelength, and that allows them to see a greater level of resolution. It's a little bit hard to sort of to think about, but because a photon is moving on a quite a large wavelength, if we're trying to look at a small object like that, this photon of light could be at any stage in its wavelength when it hits it, and we wouldn't know which stage it's in. So this would effectively, this small dot would look like a big splodge, the same size as the wavelength of light. So we basically can't see anything smaller than the wavelength of light. And that's the limiting factor with light microscopes. Electron, smaller wavelength, we could see that small object, see greater resolution. You don't really, if that didn't make any sense to you, don't worry. It's not like on the syllabus or anything. It's just kind of trying to explain why the difference is. But you do need to know the stuff in the boxes. So beams of electrons, this gives us black and white images. You can add color to these afterwards. So sometimes you see really nice electron images that have got false color added. So a computer, you can do it on a computer basically afterwards. Higher res and mag. down to around 0.1 nanometers. So a huge difference between these two. And to be honest, we actually are improving electron microscopes. So they're getting better. They're getting even more, you can see even smaller and smaller things. So yeah, I'm sure you guys have all used light microscopes. They're kind of ubiquitous in learning about biology. You won't get to use an electron microscope. Most people, even if you studied biology at university, you probably wouldn't use one in university. Maybe if you did chemistry undergrad, you'd have to be doing quite a specialized course to need to use an electron microscope. You need quite a lot of training. They're quite expensive. So yeah, most of you probably will never handle one of these in person, but we see the images all the time in our like textbooks and stuff. So let's just quickly label the light microscope. So let's throw this out for you guys. Can you identify any of these features on a light microscope? Have I ever used one? No, I never use an electron microscope. Yeah, I tried to, I don't know, the side of biology that appeals more to me is like the whole organism side, like the ecology, like animal biology, behavioral, that sort of stuff. So I tried to like avoid being in the labs as much as possible. But yeah, I think we did your thesis on something niche that you had to look at small things. Maybe you'd get to use one. Like all universities will have electron microscopes. I don't know if they let undergrads loose on them. Maybe just postgrads. Nice. So we've got our objective lenses here. These typically 5 times 5 times 10 times 25, maybe times 40. If you've got a more expensive microscope, really varies. Nice. 2 is stage, yes. So this is where we put the specimen on. So usually on a microscope slide, we put the specimen there. 3, we've got our light source. So this is quite a basic microscope. This is just it looks like it's using a mirror to reflect light. The more expensive ones, they have like a built-in light source here. So it just shines directly through. They both work, but if it has a built-in light, they're usually a bit better. Probably have a higher, a more powerful lens on the objective lenses as well, because they're more expensive. So there's a lot of variety in the quality of a microscope. The top of the range ones, you're looking at several £1,000s. So they vary. This one is the eyepiece lens. Oops. These are normally times 10 magnification. And then we've got our 2 focusing dials. So this is the coarse focus. This is for moving the large amounts. And then we've got our fine focusing lens, or dial, I guess let's say. So you move it most of the way, you move it quickly with your coarse one, and then when you got it almost, you can see roughly what you're looking at, then you use your fine focus to really get it fully in focus. And you can tell it's in focus because you can see clearly what you're looking at. So if it's blurry, you need to fiddle around with the focus. If it's dark, it's because you haven't lit it up, or because your specimen is too thick. So you can only look at very thin things with a light microscope. If you try to, I don't know, even looking at a sheet of paper, that's too thick. The light's not really going to pass through it, and it wouldn't work. So you have to have tiny, thin specimens. Yeah, that's a good thing to remember, Evan. Exactly. Olivia, depends on the specs. But yeah, I'd say you should be comfortable with these different terms of a light microscope. And you also need to know how to use them because of the required practicals. You need to be able to set one of these up and like explain how you use it. So the total magnification, this is quite a simple calculation. It's just the objective lens times the eyepiece lens. So say we were using, because you can select a few different versions of these. Say we're using our times 25 objective lens. And then we're using our eyepiece is basically always 10. We just multiply these two magnifications to give us our overall magnification. So 25 times 10 equals 250 times magnification. And that'll be our magnification with that one. If we used a 40 objective lens, we would have 400 times magnification, and so on. Relatively straightforward calculation. Nice. So let's look at how we prepare a specimen. So we said it has to be very, very thin. Otherwise, the light can't pass through it. So we generally take our specimen, we put it on a microscope slide. So this is a slide. This is a cover slip. So we take a thin specimen. Oops. On slide. We add stain. Stain adds contrast. Yeah, exactly, Timothy. So contrast, you might be familiar with contrast from like fiddling around the settings on your TV or your phone or something where it says increase or decrease contrast. It basically enables us to see details. Because we're using such a thin specimen, the light can sort of go through it very easily. So if we stain it, it will make it easier to see. If you don't stain, if you're looking at cells and you don't stain them, you can't really see very much. So it's much better to stain. Also, what we can do is stain specific things we want to look at. So Muno, exactly. If we wanted to look at DNA, we would use a stain like methylene blue, which stains the DNA. Therefore, it's going to stand out very clearly. If we were looking at plant cells, we might use iodine, which stains the cell wall, which makes it easier to see. Or it also stains the starch granules. Callie, it's not necessarily always 10, but that's like by far the most common. So for GCSE answer, I would pretty much say like basically you can say it's definitely 10, but you can swap them out and you can use different ones. But yeah, I would just go with 10. Stain, add contrast, cover slip. Oh, cover with cover slip. Probably shouldn't have written cover. Top with a cover slip. So this just helps to flatten out the specimen, making it thinner, and it protects the microscope objective lens. So it's not just like dipping into the specimen, which could cause problems. So final thing, we'll just look at these images. So these are both from electron microscopes. There's two different types of electron microscopes. We've got transmission electron microscopes, also called TEM, missed the T, and I drew a weird M. If I try and, it's because I'm rushing now to try and get done by 6:30. If I try and rush, it's like a case of more haste, less speed. So we call these TEM, we call these SEM. Essentially, all you need to know is that these have higher resolution, but only 2D images. Lower res, 3D images. So if you're looking at a flat two-dimensional picture, well, it's a big clue that it's a transmission electron microscope. If you're trying to decide whether it's a TEM or a light microscope, because they're more similar, look at the level of detail you can see. Like in this image, you can see the cell wall clearly here this is a prokaryotic cell. So you can see the cell wall and you can see all this. It's got to be an electron microscope. We wouldn't see that much detail with a light microscope. This would just be blurry. And in this animal cell here, we can see all these mitochondria. You basically can't see any organelles with a light microscope. The organelles are too small to see with light microscope. You can see like the nucleus and you can see the vacuole. That's kind of it. So something like this is definitely electron microscope. If it's an image like this where it's 3D, you know it must be a scanning electron microscope. So yeah, that's how you can interpret the images. It's worth learning those. I can't remember exactly if it's in all the specs that you need to know the difference between these two, but it's definitely in all of the A-level specs. So if you're going to carry on, you definitely need it for A-level, and I think you need it for most of the GCSE specs as well. But yeah. So you guys happy with that? There's a little bit more, but we haven't got time to do it today. So this is what a couple more questions we would have got through if we were doing a normal web class on the website, but we're going to skip those over. So yeah, basically, do you guys have any questions on anything we went through today? Like in the ones. Oh yeah. 1 times 10 to the power of 0. I like it. Super relevant. Milagros, that was good timing then. Amy, yeah, good question. We've got a couple more weeks of these GCSE classes. I think, or maybe just one more week actually now. And then then it's like on the website only. So hopefully some of you guys are going to be signing up. I think the price, I don't know if the price has been released yet, but it's a lot cheaper than the A-level classes if you guys are familiar with those or you've got siblings who do those. I think it's something in the region of 15 to 20 pounds a month, which compared to like tutoring is a total bargain. So yeah, the A-level classes are on the website. That program's been running for a few years now, so that's already ready to go. So check out the website. Especially if you're doing A-level, check it out. Like it's up and running now. The GCSE ones are starting in a couple of weeks' time. Before that, we're doing these YouTube classes. So subscribe to our YouTube channel, set a reminder. Monday is physics, Tuesday's biology, Wednesday is maths, and Thursday is chemistry, all at 5:30. You can set a reminder so you don't even have to hold that information in your brain. You can just get a notification before the class starts. Enter the competition. 10 free accounts. Got to be in it to win it, so there's no harm entering. It's not a you don't have to pay or anything, so it's a free competition, and the winners will be decided next week. So let me look at your questions. Sorry. Pay to unlock the current format for GCSE. It's a continual sort of subscription, D. So you pay a monthly fee and you can access the materials while you're subscribed, if that makes sense. So it's sort of like an ongoing thing. It's not like downloading just a pack. I mean, you could, if you sign up for a month, you could download all the learning packs. That's definitely possible, but it's more about like attending web classes and sort of like doing all the stuff live. That's the real benefit of SnapRevise, like the live and the interactiveness. So you could essentially nick the resources, but you won't be getting the classes, which are I think the best thing. It's sort of like the selling point. Do you need to learn microscopy for CIE? Yes. Yeah, it's in all the specs. It's like such a core part of biology that it's in all of them for IGCSEs as well. Timothy, that's good to hear. Signed up for one year. Nice. I didn't realize you have to sign up for like a year at a time. That's good. Straight in. Yeah, that's it. I'll stick around for a couple more minutes if you guys have got any questions. Otherwise, thanks for joining, and see you guys next week if you want to join for the final session or on the website, hopefully, if some of you guys are convinced to sign up. You can always just try it for a month and see how it goes. No worries. You're very welcome. Just joined. What time did they start? 5:30, Jess. So if you want to join next week, they start at 5:30. We've got ones with other subjects, which are running the whole time. Amy, I think it's something around that sort of price. I'm actually not 100%. I asked when we started these classes, and I think the price hadn't been set at that point. But if you maybe have a look on the website, maybe it's on there now. But I think it's in that sort of region, the price. Also, if you do more classes, it gets cheaper. At least that's how the A-level ones work. So if there's no more questions, I'm gonna ring off. So yeah, I hope to see you guys next week and on the website in the future. Have a good evening. Bye.